Okay, so what we're going to be talking about is molecular phylogenetics. Molecular phylogenetics. Can everyone see that? It's kind of cool, right? No. All right, so we're specifically going to first give an overview here. So there'll be an overview and a little bit of history. So we'll spend a little bit of time on history. And then after that, we're going to talk about how to build phylogenetic trees. To build trees. Build trees. And then how to build trees kind of falls into two different methods, which are matrix and character-based methods. And we're going to talk about how to use both of those methods today. All right. So let's go to the next slide. But what is molecular phylogenetics? So to start off with, just because I think this is a review, phylogenetics is just the study of evolutionary relationships. So this is all about evolutionary relationships. All right. And kind of phylogenetics has been happening for thousands of years. So basically, anytime that we say, okay, flies are more similar to mosquitoes than they are to humans, that is a phylogenetic analysis, and that's been happening like at least as far back as early ancient Greece, if not further. But more recently, we've been using phylogenetic trees. Phylogenetic trees. So this has kind of been since the 1800s or so. And this is how we represent phylogenetics. And I'm going to show you a tree in just a minute. By the way, for the couple of y'all that came in more recently, Chesley's out. I'm going to give a little lecture on trees. And yes. Okay. So phylogenetic trees. So first, I'm going to draw out a phylogenetic tree. Before that, here's two examples of like historical phylogenetic trees. So right here, we have one of the first phylogenetic trees ever written down. This was in one of Darwin's notebooks. And it's funny, at the very top, he says, I think. He doesn't know. And this was him like speculating about the origin of life. And then right here is a tree from SARS, from the SARS coronavirus, which is topical now. And we're actually groups of researchers are making trees like this for the current COVID-19 coronavirus. And it's a good way to track where the virus came from, how it's evolving, things like that. So I'm going to draw you a phylogenetic tree. So I'm going to draw you a phylogenetic tree. Let's say we have some animals right now. We have a worm. We have a snake. And the reason why I'm drawing this is I'm wanting to give you some terminology about trees and how we talk about trees. So here's a snake. Here's an alligator. Here's a mouse. And here's a human. And I'm going to give each one of them a dot like this. Whoop. And these dots are called, these are called leaves. So this is a leaf. A leaf. This is a leaf. And we know, for example, that mice and human are related to each other. So we draw a relationship here. And up here we have some sort of ancestral mammal. So only the leaves are alive today. And this is some mammal that no longer exists, but is an ancestor of both mouse and human. And what we call this right here is an internal node. So this is an internal node of the tree. And we can draw more internal nodes. So snake and alligator are related. This is a reptile, some ancestral reptile. Here's another internal node. And then reptiles and mammals are related. They're both vertebrates. So there's a vertebrate. So here's another internal node. And then we have this worm sitting out here by itself. And this can be connected to vertebrates. And these are all animals. So that's an ancestral animal. And what worms here are, what we call this kind of this whole wormy branch here, we call this an outgroup. So it's like it's kind of separate from the rest of the tree. And what it allows us to do is we form right here a root of the tree. So, this is a tree, so the roots are at the top, of course. And there's the root. So we basically have leaves, which are all alive today, internal nodes that are ancestors, roots, which are the furthest back ancestor, and then outgroups that are less related.
And there's no like external node?
There's not really an external node. No, basically everything's going to be connected to the root by definition here. Yeah. If it's a rooted tree, you can have different things if it's an unrooted tree, which basically just means, do we have, an oldest common ancestor here?
Coming back here and editing these, I didn't answer that question very well. Essentially, there are external nodes that I didn't mention here. And those are often, external nodes are often these leaves, just with different tree topologies. So if you want to learn more about that, go look up different cladograms and tree topologies in phylogenetic trees to learn more about that.
All right, so I drew a tree, and basically we can make trees. So let's see. We can make trees from traits and also molecular info. So in this case, we made a tree from traits, but we also could do this from molecular information like DNA, proteins, or something. And when we do that, it's called molecular phylogenetics. So that's what molecular phylogenetics is. So far, so good? All right. So now's the historical part.
Originally, this was my first time using this software, and some of these sections got deleted or not recorded. So I'm filling in here after the fact for how molecular phylogenetics began. And the answer is that for the most part, molecular phylogenetics began in 1958 with the advent of molecular information as we know it. So Francis Crick kind of early on postulated that molecular information might be able to be used for phylogenetic analysis. And then nearly 10 years later, Linus Pauling, he published a paper with more detail about how this might be done. Until 1 year later, with a paper from Eck and Dayhoff. And we know Dayhoff. This is Margaret Dayhoff, basically the founder of modern day bioinformatics, all sorts of stuff on protein alignment. And she and Eck described the evolution of ferredoxin, using amino acid sequences, using AAs. So this is a protein, an amino acid level molecular phylogenetics. And there was another big paper in '67 from Finch et al. That did a similar thing for cytochrome C. And so far, this is all amino acid level molecular phylogenetics. And a lot of that was because there was a lot of skepticism still and misunderstanding, I think, about DNA and its importance in evolution. But that changed to a certain extent in 1977 when Carl Woese and his postdoc George Fox, they released a paper where they used the 5S and 16S sequences of methanogens evolution. And from this paper, came our modern understanding of the three-domain system. We separate all life into eukarya, into bacteria, and archaea. And this was really impressive for the time. So 5S sequences, to give you some perspective, are around 120 nucleotides, which was feasible at the time with Sanger sequencing. But 16S is around 1,500 nucleotides. And that just wasn't feasible with sequencing technology at the time. So these experiments that Woese and Fox did were really impressive. They first of all had to fragment the 16S sequence. And even after that, they did these giant batches of electrophoresis in 100-liter tanks of kerosene. The kerosene is in the fuel. So these experiments would not be done today just for safety reasons, because you light a match in there and the whole building goes up in flame. But we are grateful that they happened at the time. All right, and then one other thing to mention is that molecular phylogenetics at the DNA level was also used about 10 years later. This technique called paralogous rooting, which was invented by Dayhoff up here. It was used to show that eukarya are more similar to archaea than they are to bacteria. And that was really spectacular. So this is, using a bioinformatic tool and using DNA to completely uproot how we thought about biology and biological classifications. So this is one of the first real successes of bioinformatics.
And now we get into the good stuff. How do we build a phylogenetic tree using algorithms? So all of these phylogenetic trees algorithms, they're going to start with a multiple sequence alignment. Multiple alignment. And this is MSA, which we learned about. After that, you're going to get some divergence. So on some of the methods we're going to talk about, you're going to start with this MSA, this alignment, and then you're going to build an alignment matrix. And from that matrix, you're going to have an algorithm that builds a tree. These here are called matrix methods. Those are matrix methods. In the other case, we're going to go from an MSA, and we're going to go straight to a tree. But we're going to have many trees. We're going to generate however many bunches of trees. And then from there, we're going to pick some of our favorites, or often just one. And these are called character methods. Character methods. So in this lecture, I'm mostly going to focus on matrix methods because they're really easy to do in front of you. Character methods generally involve lots of big data, so I'm going to talk about those in the end, but most of this is going to be going from MSA to matrix and then from matrix to tree. All right. Makes sense so far? Hey, y'all. So Chesley's out in Brazil. I'm talking about trees, and we're just starting to get to good stuff. So here's an overview of, so I just talked about the two main methods, but it's actually a lot more complicated than that, and we're only going to talk about some of those things today. So you can see there's kind of three big branches of how you build a phylogenetic tree. On the tree generation side over here, there's this branch right here, and this branch is what I talked about with matrix methods. And in a minute, we're going to talk about UPGMA, which is what I mentioned earlier. And we're also going to talk about the neighbor joining algorithm. And just know that there's other algorithms that exist. And then on this spot right here, these are the character-based methods. So we're going to talk about maximum parsimony here, maximum likelihood, and that's it. Then there's this kind of amorphous blob over here. And these are all about evolutionary distances. So these are models of how much distance is there between sequences. And we're going to use them on the matrix methods. We're actually going to use them to build a matrix. And we're also going to use them in the character methods because they're going to like define a goodness of fit basically for a tree. Okay. So one thing also to note is that there's lots of things that can go wrong when you're building a tree, and we're just going to talk about one of them basically. So I'm going to briefly mention long branch attraction. That's one thing that can go wrong when you're building a tree, but know that there's many others. Make sense? Questions? All right, let's build a tree. Oh wait, we have to build an alignment matrix before we can build a tree. So let's start with that. And if we're going to build an alignment matrix, I need to give you an alignment. So I'm going to give you one. So here I'll label it alignment. And how am I on time? Okay. So we're going to just do 3 sequences so that we can work these out by hand. And the first sequence here, sequence A, I'm going to say it's A, T, A, A, A. Sequence B is G, T, A, T, A. And sequence C is C, T, A, A. So there's an alignment. And now if we want to make an alignment matrix out of this, we need to do pairwise alignments of these. So I'm going to write those out. So here is, let's do sequence A versus sequence B first. So here's A, which is A, T, A, A, A. Here is B, which is G, T, A, T, A. And if we align these, we're first going to get a transition. So I'm going to write S for transition. Then these ones are the same. Then we have a transversion here, and this one's the same. On board? So now we're going to do sequence, let's do A versus C next, colon. So A is still the same, hasn't changed, A, A. C is a little different, C, T, A, A, A. We have a transversion here, and the rest are the same. And then finally, zoom in a little bit, sequence B versus C. So here, B is G, T, A, T, A. Here is C, T, A, A, A. Here we have a transition, and then here's a transversion, and there. So I'm going to leave this up here, and we're going to use it as we talk about how to make a transition matrix, an alignment matrix, which is what we're going to do right now. So the easiest way to make a matrix is going to be a Hamming distance. So we learned about Hamming distance. So this is one Hamming distance. Hamming distance. So what I'm going to define is I'm going to say we have a metric called P. And P is the alignment, pairwise alignment. And it's going to be equal to D over L, where D is my Hamming distance, and L is my length. Okay. So that's our simplest matrix, and I'm going to make it. So A, B, C, A, B, C. The distance between anything in itself is 0, and we don't need this. It's a triangular matrix. And the difference between A, let's see up here, A and B, let's see, we have 2 mutations, and we have a length of 5. So this is 2/5. A and C, there's 1, so 1/5. And B and C is 2, so 2/5. So that's a Hamming distance matrix. What's wrong about this matrix? So if I'm wanting to describe like how close 2 sequences are to each other, what might be some downsides of this sort of thing? Some of them are unnecessary, okay. What do you mean by that?
Combining A and B together will obviously be 0.
Well, that's actually interesting. So what if we had the case where we have A and we end with A, but what if a mutation did happen? What if we went through T somewhere evolutionarily? That could happen, and we wouldn't see it. So because of that, what I'm going to add here is that we're underestimating mutation. Because some mutation could happen that we didn't see. Like, it looks unnecessary from here, but evolution cares. Anything else?
C
Yeah.
has the same distance with A and B.
C has the same. Yeah, so like in this case, but I could make an example where C had a different distance, right?
Yeah.
Yeah, so that's one thing about this particular example that's different. But I don't think it'll affect our ultimate tree. Thank you.
Since you need it to be the same, though.
Yeah, I mean, we'd expect for Hamming distance. Oh, I see. So one thing on that then is that you say the distances are the same, but they're kind of different, right? Because transitions and transversions are different. I guess in this case, the example isn't as good because they both have a transversion and a transition. But one thing to note is that like right here, we're losing information about what's a transition and what's a transversion. It's like all mutations aren't created equal. So we're going to say, so we're losing mutation info. Losing information about mutation. So what we're going to do is we're going to try to form a few other measures of evolutionary closeness to fix those problems. So the first thing is that we're underestimating mutation. And there's a model that tries to fix that. Where did my notes go? Here they are. So the first model is called Jukes-Cantor. And it's often abbreviated JC. And kind of the principle that this relies upon is, let's imagine we have an adenosine. What can it do? It can mutate, so it can mutate to a T, it can mutate to a G, and it can mutate to a C, or it also can stay the same. It can go in on itself. Move this a bit. So what Jukes-Cantor is going to say, they're going to say RA is equally likely to mutate into anything else. And we're going to call that frequency alpha. So we say that all of these have alpha. You have an alpha chance to go into T, to G, or to C. But one thing we know is that our total number of nucleotides doesn't change, which means all of these must add up to 0, which means our chance of going back to our self is minus 3 alpha. So these are our probabilities. And if you play with those and you do some fancy math, you end up getting an adjusted distance function. So here's our distance for the JC model. And given this information about how A mutates, we're going to say this model is equal to minus 3/4 times the natural log of 1 minus 4/3 p. And this p here is the same p as this, let's see if I can move, as this Hamming distance p up here. So this p is the same as this p. And this model will help a little bit because it's actually going to, when you have more mutations, it's going to give a further away evolutionary distance than just Hamming distance. And it does so, like the more mutations there are, the bigger that difference is, which is going to adjust for the chances that an A would turn back into an A. So we can rewrite our matrix. So I should have made these matrices blue so we can keep track of them. So there's one of them. And now I'm going to write the Jukes-Cantor one, the adjusted one, A, B, C. So these are still 0, because if you plug in 0 to this equation, you still get 0. And we still don't care about these. And I plugged this into my calculator earlier. And for AB, we get 5, 7, 2. For C, we get 0, 2, 3, 3. And here we get 0.572. So this is the Jukes-Cantor model. So I had 2 downsides earlier. I fixed one of them, and I still need to fix the other one. What's the one that I still need to fix? What problem do we still have with Jukes-Cantor? So which of these downsides did we fix? So we had an underestimated mutation and we had a losing mutational info. Which one did we fix? Exactly. Yeah, exactly. So that's one we fixed by saying, okay, you can go back to yourself, and that's still a thing. But we still don't have anything about like mutation, about like transitions or transversions or anything. So that's a problem. So we need a new model. Let's do a new model. How's my time? Okay. doing all right. So here's a new model. So the last one was called Jukes Cantor. This one is called Chimera. I think I'm saying that right, Chimera? Yeah, Chimera. And it's Chimera 2-parameter. 2-parameter. And this is often abbreviated K2P. And this has a model just like Jukes Cantor, so I'm going to show you it like that. So let's say we have an A. An A can go to a T, it can go to a G, it can go to a C. But now we're going to take information about how, for example, A to T, that's a transversion. And A to G is a transition. And this is a transversion. So what K2P says is that if you made a transversion, that's going to be beta. And if you made a transition, that's going to be alpha. So this is beta. And then, of course, if you go back onto yourself, since this needs to equal 0, this one's minus 2 beta minus alpha to make everything balance.
Why would AC be a transversion?
Isn't AC a transfer? Did I do it wrong?
A to C.
Uh,
A to C is transversion.
yeah, OK. So that is correct. A to C and A to T are both transversion. Is that correct?
Yeah.
Yeah? OK. Cool. So that's good. I thought I looked them up, but I'm glad I did. OK. So that's how this model works. And if you end up doing the same sort of manipulations that we did at Jukes Cantor and you do it to this model, we're going to end up getting a much heftier looking equation. So here's my distance of K2P. This is going to equal to 1/2 log of 1 minus 2P. And this is a different P. I'm going to define it in a minute. Minus Q. And then we have a 1/4 log of 1 minus 2Q. And here the capital P is the fraction of transitions. And capital Q is the fraction of transversions. So here we have 2 parameters. And we can rewrite our matrix again. So here's A, B, C. A, B, C. We still have zeros. We still don't care about these. And if you plug our P from the Hamming distance in here, you get 0.255, 0.239, and 0.658. And now this distance is more reflective of transversions and transitions and the difference between them. Questions about that? So these are all ways to adjust our distances. But there's still lots of other problems. So here's two models. We went over Jukes Cantor and went over K2P. And I'm going to group all the other ones into other. And there's other assumptions that we didn't think of. So for example, what if you have an unbalanced GC content? So what if your animal or your organism has 60% GC? Well, neither of our models accounted for that, because if you have a mutation, you're more likely to get a G or C if you have more Gs and Cs around. So there's a model called HKY85 that accounts for that. You also have many cases where some nucleotides mutate more than others. So I'm going to call this like unbalanced mutation. So for example, G is more likely to mutate to C than it is to A. And for that, you need to use a gamma distribution model. So there's lots of these different models. And the more complex they get, the more likely you are to overfit your data, the more likely you are to get the wrong answer. So there's kind of a balancing act between how many of these correcting parameters do you put in and how overfit your data is in the end and how much data you have coming in. And I mentioned a few, but there are many more. And that's what this slide is for. So up here, we have, remember before I was saying what if you have like 60% GC content? Well, the 2 columns here, equal base frequency means you have a 50% GC content. Unequal means you have a not 50%. And then these columns, if you treat all mutations equally, you have one substitution type. So like here's Jukes Cantor that we just talked about. But if you have transitions and translations different, you come up to K2P. which is 2 substitution types. But if you wanted to, you could treat every mutation as its own thing, and then you get up to more substitution types or extra parameters like that. So here's some common ones, and then here's another common table, just showing it in a little different way. You have differences in phase composition and rates and things like that. Right. So those are evolutionary models. Any questions? And let's see. So do I do additive phylogeny before the break? Yeah, let's take a quick break.
That got deleted, unfortunately, after the fact. So this next step is, remember, our goal here is to go from alignment to an alignment matrix to a tree. And this section here is going to focus on that last part of going from a matrix to a tree. And we're going to do this by hand. And to go from a matrix to a tree, I'm going to need to give you a matrix. So I'm going to draw one here. Here's a chimp. Here's a human. Here's a seal. And here's a whale. And let's zoom around here. And here on the other side, oops. Here on the other side, we have chimp, human, and whale. And of course, an alignment with itself is always 0. And then we're going to have distance of 3, 6, 4, 7, 5, 2. So I'm going to take this matrix and I'm going to draw you a tree. So here's a chimp, here's a human, and I'm going to say they're connected here, and this is 1, and this is 2. And then we're going to have 3 here. Here is a seal. Here is a whale. They're connected with 2, 0. So there's a tree. This is an unrooted tree. And take a second if you need to pause and then make sure that I drew this tree correctly. Right, so I'll look at a few of these. So for example, between chimp and human, there should be a distance of 3. So here's chimp and here's human, and here's 1 and 2, so that makes 3. Between chimp and seal is 6. So chimp, here's 1, and then now we're at 4, and now we're at 6. So I think this tree works, but how did I do that? Well, I'm going to show you an algorithm to do that, and then I'm going to tell you about how that algorithm usually doesn't work. So in red here, I'm going to say algorithm, we're going to call it algorithm number 1. And this doesn't really have a proper name because it's not too complicated. So I'm just going to call it additive phylogeny because that's what one of the textbooks I was reading uses. Phylogeny. And here's how this algorithm works. The first thing we're going to do is we're going to select any node in the matrix. So I could select this 5 here, or I could select this 2 here. Then I'm going to find the edge lengths that node. Lengths between human to A and A to whale, or something like that. And I'll show you how to find that. And then our third step is to rewrite the matrix. The matrix with if I'm connecting seal and whale, I'll rewrite seal and whale as just one node, and this will make more sense as we work through it. So let's do this exact same example, and we'll hopefully end up after this example with the same tree as before. All right, so let's start with this matrix. So here I'm just going to abbreviate CHSW, CHSW, and our identities here, and we don't care about these. This is the triangular matrix. So here we have 3, 6, 4, 7, 5, 2. So I'm going to pick a node at random. This node looks cool. Let's do 2. And now I'm going to connect. So this is seal and whale. So I'm going to write seal, and it's going to be connected to whale. And I'm going to call this node right here A. Okay. And now I need to find the edge lengths between seal and A and whale. Because you know this here could be 1 and 1, or it could be 2 and 0. Those are basically our options, or 2 here and 0 here. And we're not sure which one it is. So the way to do this is this handy little trick, which says if we have some node here, so let's say we have chimp over here somewhere. And the trick is, if I want to know the distance between seal and A, what I can do is I can look at the distance between seal and chimp, and I can subtract the difference between whale and chimp, add the distance between seal and whale, and divide by 2, and I get this distance. And if you don't believe me, it may help to draw this out geometrically. If you look at all those lengths, essentially they end up becoming one circle, and everything works out. But I'm not going to prove that right here. So essentially what I want to do is the distance from seal to A, so I'll do SA, it's going to equal 1/2 times the distance, and I pick some random point. So here I picked chimp, so I'm going to say the distance between seal and chimp, and then minus the distance between whale and that same thing, so whale and chimp, plus the distance between seal and whale, and that here equals 1/2. Let's see, the distance between seal and chimp, S and C, is 6. So this is 6. And then the distance between whale and chimp is 4. And the distance between seal and whale is 2. All right. So that equals 2. Right. Is that this this branch length up here must be 2, which means that this one must be 0, right? Because the distance seal and whale is 2 total. All right, so we have that, and here's our matrix here, and we're going to rewrite it now, and we're going to rewrite it in terms of A. So here's chimp, human, and where seal and whale was, I'm just going to have A. So here's chimp, human, and A. Our identities are still 0, and this is still a symmetric matrix. And we knew the distance between chimp and human. That hasn't changed. It's still 3. Now to find the distance between chimp and A, so we know that, for example, chimp and seal were 6 apart, and A is 2 away from seal, which means the distance between chimp and A must be 6 minus 2, or 4. Same thing with the distance between human and A. So human was 7 away from seal, so now it's 5 away from A. So there's our new matrix. We're going to do the same thing as before, where we pick some random connection. So I'm going to pick between chimp and human, and now I'm going to draw that one. So here we have chimp. Oh, let's do this. Chimp, and I'm going to connect chimp to human, and I'm going to call this node here B. And now, just like before, we need to find this distance. So the distance between chimp and B is equal to 1/2 of the distance between chimp and some random point. So I'm going to pick A. So chimp and A, minus the distance between human and that random point. So here's human and A, plus the distance between chimp and human. So this is going to equal 1/2, and here is 4 minus 5 plus 3, and that equals 1. So here's 1. Between chimp and B must be 1. And since the total distance here is 3, This one must be 2. Algorithm, we would rewrite this matrix one last time with just A and B. But this is a little easier because, see, we have here A with seal and whale and 2, 0. And we know that the distance between chimp and seal in total is 6, right? We know that from this original matrix up here, this 6 right here. And since we have a 1 here and a 2 here, then this connector here that connects B and A must be 3. And now let's check. So here, up here is my original tree, and I think it matches this new tree that we just made by this algorithm. So the problem with this works with additive trees. Well, what is an additive tree? Well, an additive tree is a tree that this algorithm works with. So you'll see in a minute, I can make a different matrix. And if we try to do this algorithm with some matrices, we'll get trees that just don't make sense. Like they'll have negative edge lengths, for example, which doesn't make sense. So additive trees are trees that work with this algorithm. And if a tree is not additive, this algorithm does not work. And one thing that's good about this, so what's good about this, and I'm not going to prove today, but what's good about this is that if a tree is additive, then this algorithm gives just one solution. Because we have just one solution. And then that's with the caveat that it's one tree that has all its nodes have degree of 1 or 3. So what I mean by that is, like, if we look at this tree up here, presumably we could put, an extra node here and call it Z, and, have here be 1.5 and here be 1.5, and we could do that forever. And notice this node would have a degree of 2, meaning it's connected to 2 things. So this additive phylogeny gives a solution, a one possible solution, where all the nodes have degrees of 3, like they are here. See, this is connected to 3, or 1, like seal is just connected to 1 here. So that's what's good about this algorithm. There we go. Now coming from the future again, since this was deleted, is what happens if we have a non-additive tree. So I'm going to give you an example. So using the same sort of matrix as before, we have chimp, human, seal, whale. Here's chimp, here's our zeros. I'm going to modify the matrix just a little bit. I'm going to call this one 4, 4, 8, 4, 8, 2. And this one's not going to work with the previous algorithm. And I'm going to show you really quick. So I'm going to pick a random connection here, just like before, between seal and whale. So here is seal, here is A, here is whale, and I want to know what the distance between seal and A is. So I'm going to do 1/2, and I'm going to do the distance between, just like before, seal and chimp, minus the distance between whale and chimp, plus the distance between seal and whale. And that will equal 1/2, 4 minus 8 plus 2, which equals -1/2. And so we can't have negative edge lengths. These need to be positive. So this matrix doesn't work. So we need a new algorithm. And that new algorithm has a very lengthy name. So let me scroll down here. So this new algorithm unweighted pair group this again. And no one wants to write this again, so we just call it UPGMA, and we advise the people who made the algorithm to come up with a better name next time. So let me show you how this algorithm works. So I'm going to have the same matrix as before, and I'm going to abbreviate C, H, S, W, C, H, S. And notice we actually don't need to write the W here. If we go up to this original matrix, you'll notice that this last row is basically completely redundant besides the 0, and that 0 is always going to be there. So often when people write these matrices, they don't write the last row. And we know that these are zeros here. We're going to take the same matrix as before. So 4, 4, 8, 4, 8, 2. And for this algorithm, we're going to pick a node just like before, but we're required to pick the smallest node. So I'm going to pick this one. I'm required to pick this one. Before I happened to pick it, but I could have picked whatever I wanted. Here I need to pick it. And just like before, we're going to connect these. So here is, let's do this in black. Here is seal. Here is whale. And we're going to connect them to assign the edge lengths. We're essentially going to say that this value, this node right here, is going to have a certain distance from all the leaves. And that's a requirement of this algorithm, that we assume that, well, you'll see that the root has the same length from all the leaves. But we're going to say that this node has a distance of 1/2 of this number. So 1/2 of that distance. So this must have a distance of 1 from the leaves. If this node has a distance of 1 from all the leaves, then this length must be 1 and this length must be 1. And that works, right? Because we said that seal is 2 away from whale, and here seal is 2 away from whale. So we do that, and now we're going to rewrite the matrix. Let's zoom out, and we're going to rewrite the matrix. C, H, and before I called the union of seal and whale A, but that's not really proper. In reality, it's a set, and it's a set of seal and whale, so that's how I'm going to write it. And then we don't need the last row, so we just need chimp and human. These are 0. We don't care about this one. Between chimp and human is still 4. As far as calculating this distance, it's just the average of the previous ones. So chimp was 4 away from seal and 8 away from whale. So for the union, it's the average. So this is 6. So it's going to be 6 away here. And for human, the values happen to be the same, which makes this a little easier. So this is also 6. Repeat. So I'm going to pick the smallest one, which is chimp to human, and I'm going to write that out. And for the sake of not rewriting things, I'm going to write it over here next to the other one. So here is connected to a node. Now this node, see our length is 4, so this node is half of that, so this must be 2, which means each of these have 2. Now my matrix again, so this time I'm going to rewrite it over here. And now I have the union of chimp and human, and I have the union of seal and whale. And then I just have to write one of these over here, C and H. And this is 0, just like before. And then the average for chimp is our 6, because they're both 6 away from seal whale. Chimp is 6 away from seal and whale, and human is 6 away from seal and whale. So the chimp-human union must be 6 away as well. And now we do the same thing on this new matrix. So here's the smallest value, 6, and that's going to connect the seal and whale and chimp and human clusters. So it's going to connect these here. And this is 6, so this root right here is going to be 3 away from all of the leaves. So notice previously we just split this, so but this is not going to be 1/2 and 1/2 because this is 3 away from a leaf. So this value right here needs to be 2. Needs to be 2, so that this node here goes 2 and then 1. So it's 3 away from this leaf, and then 2 and then 1, 3 away from this leaf. But over here, it needs to be 1, because 3, we go 1 and then 2. So 3 to get to this leaf, 1 and then 2 to get to this leaf. And there we go. There's our tree. Here's our root. Here's one of our leaves. This is a leaf as well, right? And there we go. And this tree has a property that says it's ultrametric. I skipped this part in the original lecture, so I'm going to repeat it in a later slide. But that means that the distance for this tree up here, the distance between the root and all the leaves is always 3. This property means that we're assuming a molecular clock. Assume molecular clock. In other words, we're assuming that everything evolves at a constant rate. Right? If everything's the same distance from the earliest ancestor, then everything evolved at the same rate, which of course is not always true. Things that reproduce faster are going to evolve faster just because they have more mutations when they divide or reproduce. This also has an additional problem, which maybe I'll talk about in the next slide. I'm not sure. If I do, you can skip that. But the additional problem is that let's imagine we have a case where we have a correct tree here. And this correct tree looks a little bit like this. If we apply this algorithm, the algorithm is going to say, okay, 2 and 3 are really close together. So this is UPGMA. It's going to say 2 and 3 are really close together, so we're going to group 2 and 3. And then 4 is a little closer still, so we're going to put 4 here. And then 1 is left over, so we're going to put 1 here. And of course, this is the incorrect tree. So this is a problem with UPGMA. And another problem is that it doesn't work with additive trees. So I guess I should write that down. So doesn't work with additive trees. Or it's not guaranteed to work, which kind of sucks because we just showed an algorithm that works every time for additive trees. But if you do this algorithm and you happen to have an additive tree, you're going to probably get the wrong answer. So that's the thing. But what's good about this algorithm is it's really fast. And oh, by the way, this phenomena here, where shorter branches go together and longer branches go together, it's called long branch attraction. This is long branch attraction. It could be called short branch attraction or similar length branch attraction. And I think I covered this in the next slide, actually, so sorry, you can skip over that. Done.
It must have stopped recording after the break. Oh well, I will re-record it at some point. So there's a few things that I took from Chesley's old slides that are UPGMA. So the first thing is that UPGMA assumes a molecular clock. So that means that all animals, all organisms in this tree must be evolving at the same rate. And we know that's false. We know that bacteria evolve faster than humans because they're reproducing more. But that's an assumption. So we see all sorts of different reasons why the clock might be destroyed. On top of that, it's very sensitive to evolutionary rates, and it only works if the data is ultrametric. So what ultrametric just means is all leaves are equidistant from root. That's what that means. So it assumes that, and that's not always the case. Advantages, this is super fast, this is super well established, and it works pretty well a lot of the times. All right. Still making the time, not bad. Let me show you where one big spot UPGMA fails. So let's say we have the following correct tree. So here's our correct tree. And it looks like this. So we have 1 here, we have 2 here, and here we have 4 and 3. So with this algorithm, with UPGMA, it's going to say, okay, 2 and 3 are really close together. So I'm going to take 2 and 3, and I'm going to cluster those. So here's the UPGMA tree. It's going to say, okay, 2 and 3 are close together. I'm going to group them. And then it'll show, okay, 4 is a little further away. So I'm going to put 4. And then even further away is 1. So I'm going to put 1. And I drew it a little wrong, but these are all equidistant. So if I erase this up here, these are all the same distance. So this is wrong. And this phenomena where short branches group together and long branches group together is called long branch attraction. Long branch attraction. It could also be called short branch attraction. Same length branch attraction. Okay, really quick for neighbor joining now. So how do we fix that? And the way to fix it is an algorithm called neighbor joining. Or not fix it, but make it a little better. Neighbor joining. So neighbor joining was published in 1987 by Satow and Ney. And this is one of the most highly cited papers of all time. So this algorithm has around 57,000 citations, so like top 20 most cited paper ever. And what it essentially does is it says do clustering basically just like you did before. But before we do that, we're going to adjust the matrix. We're going to basically normalize it on branch length. Well, it's really hard to fix, so I don't have time to draw it all out. But here's an example of how that might happen. So here's a normal distance matrix that we started out before. And then here are the sums of all the rows. So 5 plus 4 plus 9 plus 8 equals 26. Right? And the same thing here, but of course he also adds the ones that aren't included here. And then we get this length. And to create this matrix, what we're going to do is we're going to take 3 times the entry here. And then we're going to subtract, so this is AB. So we're going to take 15. We're going to subtract the A1, which is 26, and subtract the B1, which is 29, and then we're going to get negative 40. And this is a magic trick that somehow does 2 things. One, it makes the problem of long branch attraction better. It's not fixed, but it's a little better. The second thing is that this algorithm works on additive matrices too. UPGMA doesn't. If you do this algorithm on an additive matrix, you will get the correct matrix. And that wasn't proved actually for another 10 years after this algorithm was released. So it's actually really hard to prove. Questions? All right, so that's it for matrix methods. And we have 25 minutes left, so that's not bad. Oh, actually ahead of schedule, so I can spend a little more time on this. Oh, I didn't define ultrametric for you. Oh yeah, I did. I just find it as it's all the same. All your leaves are the same length from the root. What other things do we have? So you see here, this is a slide from Chesley, and he mentions that if the data is additive, neighbor joining will identify the correct tree. Otherwise, it might not work. Generally, you'll want to use neighbor joining more than UPGMA. The literature still, a lot of the literature uses UPGMA. And it's correct in some circumstances, but not most. A lot of that's just historical. Like, neighbor joining wasn't proved until 20 years ago. So it's science is a little slow. And even with our corrections that we talked about before, these distance calculations to make the matrix are almost always still underestimated. And that's kind of the way that we want it to be usually, so we don't have too crazy numbers. Another thing about distance-based metrics is that you're getting one tree. All the algorithms we talked about, you get one tree, which means I don't have any confidence estimate on that tree. Like if you ask me, how sure are you that this tree is real? I don't know. There's no p-value. There's no associated thing. So for that, we're going to have to generate lots of trees, which is what we're about to do. We're actually not going to do it because it takes a long time to make lots of trees. But we're going to go over the methods. And you can all draw you can all perform UPGMA now. That was one of the things I asked. Not everyone was in here at first, but I asked if anyone had like drawn a tree with UPGMA before by hand. And I don't think people had, which is why I thought this lecture would be fun. And these are used. So like right now, I was reading a paper about clustering the coronavirus, and people were using neighbor joining to form trees on that. All right, so next we're going to talk about character methods. Woo! So these, we go from alignment, and we go straight to a tray, to a tree. And these are going to work as follows. First, we are going to generate a tree. And we'll often do this by a matrix method. We could just put them randomly, but a lot of times in practice you use a matrix method to make this a starting tree. Then you evaluate the tree. And you'll do this by different metrics. So we could use parsimony, and we're going to talk about these, parsimony. We could use likelihood. There's different methods that we can evaluate the tree on. See how good is this tree? And then we're going to generate a new tree. And most of this time, this tree isn't a brand new tree, but it's some permutation of the original one. So I just mentioned that the new tree is a permutation. And you wonder, why would I do that and not just generate a whole new tree? And here's the reason. So there's this formula for the number of trees you have versus how many sequences are in your alignment. And that's called T of n. And the equation looks like this. Whoop. 2 to the n minus 3 factorial. And what this translates to is that if you have 5 sequences in your alignment, you have 15 trees. If you have 10 sequences, you have 2,027,025 trees. So this number explodes pretty quick. And that's the problem. Like, I can't generate all the trees, even if I have a dozen sequences. I need to have some sort of smart method to rearrange the tree and find new ones. I'm going to talk about a couple of those methods, and then we'll talk about the actual tree making. So here is tree generation methods. Tree generation methods. So first, we're always going to follow this general principle, and it's called branch and bound. So these trees are usually, like, remember when we did neighbor joining, we had subtrees and then we made a bigger tree? So these trees are usually, you start with subtrees and you grow them. And what this says is that if you have a subtree, and that subtree is worse than a full tree, you throw it out. Because if you add more parts into a tree, your score is always going to get worse. So that's a general principle. And then the more specific techniques include things like nearest neighbor interchange. This is abbreviated N and I. And then this just says you take like 2 edges that are close to each other, 2 branches, you just switch them. Now you have a new tree. You have subtree pruning, and regrafting. This is SPR. And this is where you just take a whole subtree, you grab it off of the tree, and you put it somewhere else. And you get a new tree. The next thing is tree bisection and reconnection. TBR. It's the same thing, but you cut the tree in half, flip it. So these are some things we can do to generate new trees. Questions? Yeah, this is just it. So this was next. Okay. So now the last part, which is most relevant, I think, to your outside readings and to your lab, is these metrics. So I said earlier that you need to evaluate the tree somehow. And now we're going to talk about tree evaluation. So here is tree evaluation. So the first thing you can evaluate a tree on is parsimony. So parsimony is just the number of mutations it takes to get from one sequence to another. So remember here we're going directly from alignment to a tree. There's no matrix. So how many mutations do you need to make to go from one to another, one sequence to another? And as an example, if I went ATC to ATT, this has a parsimony score of 1. Parsimony score of 1. But if instead I started as ATC, went through ATG, and then got to ATT, this is 1, 1. This is a parsimony score of 2. This may be a bit misleading. You can have very high parsimony scores and only one path, just an example. So here we would say, which of these trees is better? Well, tree 1 is better because it is the most parsimonious. Not sure if I spelled that right. No I? Parsimonious. So the method that uses this is called maximum parsimony. Maximum.
Parsimony is the one that has the least.
Yeah, that's kind of confusing, right? You have more parsimony if you have fewer mutations. Yeah.
Like parsimony means frugal, right? In other
Yeah.
words.
Oh, yeah, I guess so, yeah.
Yes.
I mean, maybe, I think the actual word might be closer to like harmonious. I think parsimony is almost like how much do they agree?
Yeah.
Like,
So the more
yeah.
frugal it is, the more parsimonious it is. So
Exactly. We want to make as few mutations as possible.
The more
Yeah, and I think the idea here is that like nature is lazy. Nature wants to make as few mutations as possible to get to its end point. So this is like an okay method to judge our tree on. This is maximum parsimony. Still good, okay. The next is maximum likelihood. So for maximum likelihood, we're going to select an evolutionary model. So remember earlier we talked about JC, we talked about K2P, talked about HKY85. These are all models. And we're going to select one of them, the one that we think works best for our data. And then we're going to ask for each tree, what's the probability, so for each tree T, what's the probability that T fits that model? So it's just a frequentist, like typical statistics. How does this tree fit? And I grabbed, do I have? Yeah, a few lecture slides from Chesley, because he's good at explaining this. He wanted to emphasize the importance of picking a model. So remember I said you're choosing a model and it really matters. So imagine if your like probability was like whether a coin flip is heads. If you're using like a normal coin, so if your model is like a normal model, your probability is 0.5. But if you're using a weighted coin, your probability might be 0.8. So the model, like the probability is completely dependent on the model. The next thing he wanted to get across, he was actually showing the likelihood. So I didn't go through all the steps, but essentially we're going to get the likelihood of a tree. We like calculate the likelihood of each site, and then we sum up all the site values. And then now we have like all of the trees that we generated, and we have a probability for each tree. And we're just going to choose the tree with the highest likelihood. Yeah. So of course we're going to have like typical problems that we have when we're doing, Monte Carlo sort of like things like this. We've got to think about, are we generating the right sorts of trees? Are we falling in local minima, like where we have a high probability, but like a high likelihood, but we're not at the highest one? Things like that. But generally, this is just a typical frequentist statistics model of trees. Cool. So what slide did I have? Yeah, I had a blank slide, which is good. Last but not least is Bayesian models. Let's use red. Before. Yeah. So there we go. So Bayesian models. B-Y-E-S. I can never spell Bayesian. That's why I have notes with me. I think that's correct. So this is what the software you're going to be using for lab uses. These are Bayesian models. And we're going to do basically the same thing as before, but for every tree, we're going to calculate the posterior probability. So this is, and the way that you denote this is you say the probability of a new tree given these values. So this here is our model, like JC, and here is like our distribution. So this is like all prior trees. And this also incorporates like prior knowledge. So some of these like distributions you'll have like, I know that these trees are better, so I'm going to say that these trees should be weighted differently. But that's the idea. So our metric for success, instead of being like a frequentist likelihood, is a probability given our prior distribution. So the way that this works, since you'll be interacting with these a little bit more, and these are probably the most complicated tree building models, is that we're going to calculate this new tree, and we're going to say if our prior here, our prior distribution, so if this is greater than our previous one, and the previous one is just the t without the star, so it's p t naught star delta here, and sometimes these evolution models are different, so you put a star too. So if that's the case, then you're going to add this tree to the distribution. So we're going to add tree to the distribution. Otherwise, we will add the tree with a certain probability. And that probability is this hunkin' thing. So here's the probability of our new tree over the probability of our priors. And this is going to equal what we just calculated with this given d multiplied by, need more space, this given t given our new model over the probability of our original tree, probability of the distribution given our original tree and our parameter. And this hunkin' thing is derived, so this is derived from Bayes' theorem. So, you know, the typical probability of B given A times the probability of A over the probability of B. So if you start with Bayes' theorem and you start applying it to trees, you'll end up getting that big old thing there. Problems with this. Calculating a posterior probability is hard. Like, there's a hunkin' equation there. We're going over tons of trees. This is like an exponential function. So the problem with this is it's computationally intensive. It's a little bit less computationally intensive if you use a molecular clock assumption like before. But usually we don't want to do that, so it's especially computationally intensive without a molecular clock. So for a long time, these methods they've been theorized, but you couldn't really use them very much. More recently, these methods for calculating posterior probabilities called Markov chain Monte Carlo methods, MC methods were released. And that's the way that we calculate posterior probabilities more quickly today. So one of the first Bayesian softwares was Mr. Bayes, which you may have heard of, and then the one Beast, which you're going to use in the lab, incorporated Markov chain Monte Carlos into its tree building. And because of that, it has like 10,000 citations. It's like super used. One of the best softwares. So that is how you build a Bayesian tree. Review a little bit. I had another picture somewhere. I don't know. I must have forgotten to put it in. But here's kind of a review of a bunch of different algorithms. We talked about UPGMA. You end up getting an ultrametric tree. You don't get scores. So all the things that I just talked about, where you go straight from an alignment to a tree, since we're generating a ton of trees, we can give a probability, especially for like something like likelihood. We're always calculating probability anyways. So we can put a p-value to a tree, and that's really good for comparing them. On top of that, what I didn't talk about as much is that generally we like rooted trees, because like from an evolutionary perspective, you want to find the latest common ancestor. And a lot of these methods, they don't give you a rooted tree by default. Like when we were generating trees at first, I wasn't making them rooted. And there's an extra step. Essentially, you add an outgroup, and then if you add an outgroup, you can root the tree, but that sometimes you lose some power, and it takes a little while. So these are methods. We've got different implementations that you might see. And that's it. Any questions? And if there are no questions, feel free.